Good morning, everyone. Honorable Professor Shyi-Tien, Actual Advisor from National Kaohsiung University of Science and Technology, Dr. Fera Kurniadewi, M.Si, Advisor from UNJ, and all lecturers and all students. Welcome back to all honorable attendants at Research Internship Seminar PKL on Thursday, January 4th, 2024. My name is Ananda Indah, and I will be the moderator today. Okay, to all speakers today, Anisa Fitriyanti, Selvia Ningsih, and Wulan Kharisma. The screen is yours. Thank you.
Okay, thank you, Ananda. Can we have to share the PowerPoint from Puan?
Is there any problem?
Okay, thank you, Puan. I want to ask permission to start now. Ya, Assalamualaikum warahmatullahi wabarakatuh. Good morning, Prof. Shyi-Tien, Mrs. Dr. Fera Kurniadewi, M.Si, and all attendees. Today, we will present about our research internship at National Kaohsiung University of Science and Technology, and the title is Use of Black Algae for Searching Valuable Substances and Energy Production. And the presenters today are Anisa Fitriyanti, Selvia Ningsih, Wulan Kharisma Dera, and this presentation is supported by Isan Lin and Yvonne. Honorable for our advisor are Professor Shyi-Tien from National Kaohsiung University of Science and Technology, and also Mrs. Dr. Fera Kurniadewi, M.Si from Universitas Negeri Jakarta. Okay, next. This is our team. Next. First of all, we will introduce about National Kaohsiung University of Science and Technology, or NKUST. NKUST is the top university in Taiwan and became the institution in 2018 and the biggest university for science and technology, merged from National Kaohsiung First University of Science and Technology, and then National Kaohsiung University of Applied Sciences, and then National Kaohsiung Marine University. And NKUST has 5 campuses with different areas. The main campus is NKUST First Campus, which is located at number 1 University Road, Yanqiao District, Kaohsiung City, 824005, Taiwan, ROZ. And then in the right side of this slide are photos from NKUST campus area. Okay, next. This is the map of National Kaohsiung University of Science and Technology for the first campus. And we will notice the 3 places here. The first is Blank Still Lake, which is the place we take a sample there. And this lake is close to female dormitory where we stayed while in Taiwan. And then we did a research internship at a biomolecule-based technology laboratory in College of Engineering. Okay, next. We also want to introduce about Agro-Environment Testing and Inspection Center, or AETIC. AETIC is one of the research center in College of Engineering, National Kaohsiung University of Science and Technology, and the center is chaired by Professor Shyi-Tien Chen and to assess for the farmers to acknowledge their planting environment, and the second one is the public to realize their living environment, and the third one, the industries meet regulatory standards. Okay, next. This internship supported by Taiwan Experience Education Program, or TEEP, and TEP managed by Taiwan's Ministry of Education and launched in 2015. And this program give a maximum stipend of 15,000 NTD per month, and TEP participants will be placed in a laboratory, research unit, enterprise, or elementary or secondary school, and provided with opportunities to attend seminars, Chinese language courses, and cultural activities to enhance their professional capabilities. And below is the website of TEP. Okay, next. And this is our general internship activities. We have three main activities. For the first is internal meeting in every Monday at 8 a.m. until 9 a.m. We discuss everything about LG topic projects and our undergraduate thesis topic in campus. And then we have lab activity from Monday to Friday at 10 a.m. until 5 p.m. We did a practicum at BBTL regarding the LG project, and we do a lab meeting every Friday at 10 a.m. until 2 p.m. And for lab meeting divided into two sessions, for English and Chinese session, the present and discuss the results of the article review with Professor and BBTL friends. BBTL is Biomolecule-Based Technology Laboratory. Okay, next. This slide is timeline internship that we did on July to October. For July, we welcoming party, introduction presentation about ourselves, and then literature review about our practice topic. And in August, we do a literature review presentation, our practice topic, and then introduction and laboratory training, determine and design internship topic, and then literature review presentation on the topic of LG, and then take something from the lake at NKUST for the first sample. And on September, we make agar medium and then sampling from the lake at NKUST for the second sample, and then isolate and cultivation the algae from the lake, and we do analysis for total nitrogen and total phosphorus and lipid, and then we added nutrient in lake sample, and then cultivation the algae from Isan sample. And in October, we did the experiments about Blank Still Lake sample, and then demonstration of the use TGA and FTIR instruments, and then we analyze lipid extraction, and then we do extraction for LG ingredients, and in the middle of October, we have a farewell party. Okay, next. Back to our research topic, and this slide is about algae in lakes and reservoirs. Algae are a diverse of aquatic organisms that have the ability to conduct photosynthesis that grow in a variety of environments such as lakes and then rivers, seas, and even sewage. Generally, algae can be categorized as either macroalgae, microalgae, or filamentous algae. And then the abundant types of contaminants discharged from urban, agricultural, industrial activities, and the others make algal blooms. And then algal blooms are dangerous because they create numerous environmental and safety threats. Okay, next. Next. This is the scheme for causing, sorry, previous slide. Previous slide. Again, the previous slide. Okay, thank you. This is the no, for causing algal bloom. Thank you. This is the scheme for causing algal blooms. Algal can grow with the nutrients. Most of the nutrient sources of eutrophication come from forest runoff and then urbanization runoff, livestock runoff, agricultural runoff that contain pesticide, and residential runoff that maybe contain detergent and synthetic fertilizer, all contain high level of phosphate and nitrogen. When the concentration of nitrogen compounds and phosphates in the water increase and helped by sunlight, algae can reproduce in large number. It's called algal blooms. Because algae blooms are also accompanied by massive death, algae corpse will us up the oxygen, so the oxygen in the water will decrease during the process of decay and decomposition, and then the fish will die of suffocation and the aquatic ecosystems will be disturbed. Okay, next. And this is the information about algal blooms in Taiwan reservoirs from Lins 2023, including the eutrophic, mesotrophic, and oligotrophic areas. Okay, next. Then the algae have various applications as a precursor for industrial products and food industry, and then pharmaceuticals, and then dyes. Next.
Okay, so on the next slide
Okay.
is about algae structure in general. So there are mitochondrion, lipid droplets, Golgi body, and others as been shown in this figure. And these cytoplasmic organelles are shielded by the cell wall and plasma membrane, which is required for photosynthesis. And what are we gonna highlight in this study is all about lipid. So lipid in general are important nutritional components that contain both long chain and omega-3 polyunsaturated fatty acids, and they also serve as precursor for the synthesis of biodiesel from PAG. Next. Okay, so the next one is some known algae. The first one is about Chlorella vulgaris. So Chlorella vulgaris is a single-celled algae species, and it's just a common little green cell wall with a thick size. It's about 100 until 200 nanometer. And for Chlorella vulgaris is a common eukaryotic microalgae species found in various natural and engineered freshwater and soil habitats. And Chlorella vulgaris also known as a healthy food to humans and used for nutrient-rich feed for aquatic animals. And among other microalgae species, Chlorella vulgaris has the highest lipid content of any microalgae, ranging from 10 to 97%. Next. So here are the cell wall structure in Chlorella vulgaris. So Chlorella vulgaris has a unimolecular structure consisting of an inner layer and an outer layer with high-like fibers. And the cell wall of Chlorella vulgaris is mainly composed of cellulose, hemicellulose, monosaccharides, lipid, protein, and glycosyl. Next. So the second one, some known algae, is about Haematococcus pluvialis, or well known as chlorophyta, is a unicellular freshwater algae with a complex life cycle. Normally, cells are green in cold season, but under stress condition or high temperature, they create thicker cell walls and change into orange-red, as in shown in that figure. And Haematococcus pluviolis is well known as the cell factory for natural astaxanthin, known as the king of antioxidant, which can be used as medicine, food coloring, and cosmetic. Next. So here are the cell wall structure in Haematococcus pluviolis. In generally, Haematococcus pluviolis cell wall has a multi-layer structure, including a primary layer on the outside, followed by three laminar sheet, a secondary layer, and a tertiary layer wall. And inside that internal space, there are any lipid droplets. Next. The next one is some non-bacteria like algae. The name is spirulina. Spirulina is a single cell organism as been shown in that microscopic view. And spirulina is a type of cyanobacteria, which because spirulina turns sunlight into micronutrients that affect high nutritional value for supporting life and providing energy. And spirulina has benefit applications such as food, biofuel, health benefits, and beauty products. Next. So here are the cell wall structure in spirulina, and its cell wall built from sugar and protein complex called peptidoglycan. Next.
In this slide, there are some ingredients inside algae. Some algae called chlorophyllarides have a bioactive compound called a lipid, and this lipid can be used as a biodiesel. Next. There are factors that affecting algal growth or cultivation. The factors are like this: light can increase the algal growth if the intensity of the light is high, and then the pH. The ideal pH of most algal species was determined to be between 7 to 9, and the extreme pH negatively affect the growth of microalgae. And then nitrogen, this nitrogen has a substantial influence in microalgal development and biochemical composition. And nitrogen deficiency in cultivation medium produce the decrease in growth and increase in lipid productivities. Next. And then phosphorus. The optimal phosphor content for the microalgae is between 0.001 to 0.179 gram per liter. And then the temperature. The ideal temperature of the microalgae cultivation spans from the 15 to 30 degrees Celsius. And above this temperature, the microalgal cell can be damaged or dead. And the last one is carbon dioxide. If in low temperatures, the carbon dioxide solubility increase, and height of carbon dioxide dissolve stimulate the rapid growth of the algae. Next.
Okay, so here are methods of extracting lipid. So there are various methods for extracting algae with all their advantage and disadvantage as in shown in the table. And therefore, to extract the algal component in the study, in accordance with Isan's research, the ultrasonic wave extraction method was used combined with acetone or n-hexane solvent because of its high extraction ability. Next.
This is the obtained ingredient varied at different extraction condition. The target of compound is an research is neophytadiene and hexadecan 1,1-bis-deoxy. And for the neophytadiene extraction, the factors of waiting time and sonication were the important factors. And for the hexadecan, the addition of hydrogen peroxide was an important factor. It can be shown in the diagrams. There are positive feedback from the waiting time and sonication in neophytadiene in extraction. And for hexadecan, we can see that the positive feedback from the addition of peroxide. Next. So this is the processing of the residue after the algae extracted. The first thing we do is isolate the microalgae from the lake, and then after that we can cultivate it, and then we can extract it with ultrasonication and hydrogen peroxide. And after the extraction, we can have a lipid, and this lipid can be used as a biodiesel. And this extraction have a algae residue, and this algae residue can be reused and recover the nutrients. And this residue used as cultivation the flora vulgaris even more. And the silker goes again. Next.
And here are the example of other aquacultural systems. For microbial aquaculture are two or more interesting microbial population that can be found in many diverse environment. For this example, use microalgae and bacteria. For the first, use Chlorella vulgaris and microcystic aeruginosa, and then Chlorella vulgaris and microorganism from original swine wastewater. Next. For the third one is use Chlorella vulgaris and Pseudomonas butida, and one of the result for this experiment, the co-culture exhibited the removal efficiencies of 78 until 85% for nitrogen, 54 until 65% for phosphorus, and about 80-60% for COD or chemical oxygen demand. Okay, next.
This is the case substance for growing algae and bacteria. The case substance are phosphor, nitrogen, and carbon source, and oxygen. For aerobic bacteria, need 1 gram phosphorus, 5 gram nitrogen, and 100 gram BOD or biochemical oxygen demand. And these aerobes bacteria need an oxygen to grow. And for anaerobes bacteria needs 1 milligram per liter phosphorus, 5 milligram per liter nitrogen, and 200 milligram per liter chemical oxygen demand. And the anaerobes bacteria doesn't need oxygen to grow. And for the algae require 1 milligram per liter phosphorus, 16 milligram per liter nitrogen, and 106 milligram per liter chemical oxygen demand. And algae needs oxygen to grow. Next. So this is the design thinking of a bacterium alga co-culture system. In this co-culture system, we use light light from the sunlight to photosynthesis the microalgae, and we added the anaerobic bacteria to break down the algae residue and release the nitrogen, phosphorus, methane, carbon dioxide to the reactor. And then we have an anoxic layer. This anoxic layer is from methane and oxygen reaction. And then the microalgae photosynthesis produce oxygen to the environment, or the oxygen going through the reactor. Next.
So here are the study objectives in the study. The first one is to isolate some algae species from a lake reservoir, and the second one to extract and discover the main ingredients of the isolated algae species. And the third one is to employ an algae bacterium co-culture system for obtaining target substances and processing the extracted algal residues. But because our internship period is only 3 months and we don't have enough time to complete the third objective, so Yvonne is a master student there, and she will continue the third objective as her own thesis. Next. So here are the experimental scheme. And the first one is algae purification and culture. And the second one is to quantify and analyze valuable ingredients via algalysis. And the third one is use the anaerobic sludge to cultivate Chlorella vulgaris. Next.
Yeah, there are material and reagent used. For the first is algae. The source from Backwing Steel Lake and KUST Kaohsiung City, Taiwan. For the application, for algalysis and valuable component extraction, and then water soluble fertilizer and an aerobic sludge. But for anaerobic sludge, we didn't do this material because of the limit internship time. This is for cultivate algae, and then we use acetone, and then hexane for extraction solvent, and then hydrogen peroxide for algalysis. Next. And we also use sulfuric acid, and then natrium chloride, and then hydrochloric acid, and then methanol, and then sodium hydroxide, and then 2,4,6-tribromofanol for algae extraction and trans-esterification process. Next. For the first we do for algae is isolation or purification. For research, the fresh algae from Backwing Steel Lake at NKUST, and we make agar medium and then stirred until dissolved, and we do autoclave for sterilization and pour 200 microliters into petri dish in laminar 8 flow, and wait until see the green colonies and observe by microscope, and cultivating the selected algae in 500 milliliters. Next.
So here are the algae cultivation procedure. Actually, the procedure is the same with the previous one, but at the end of the step is a co-culture scheme, which will be continued by Yvonne as her own thesis. Next. Okay, the next one is algae cell wall lysis. A total of 0.1 gram of algae was weighed, and 5% hydrogen peroxide was added to oxidize it. After that, it was sonicated using ultrasonic waves on a sonicator instrument to break the algae cell wall. Next.
And the next one is ingredient analytical procedures. The listed sample from the previous procedure was taken 0.1 gram and then added the solvent to the tube, and then we sonic water it, and then we centrifuge for 10 minutes, and the sample was moved to the sample bottle, and with this 1,425 microliter extraction sample and 75 microliter standard 246 TBP, and then we analyze it with GC mass. Next. And then we do total nitrogen and total phosphorus experiment. And the first one is the prepare the sample. 10 milliliter of sample was filtered, and then we take 0.1 milliliter sample and added 9.9 DI water, and then we added the hydrogen peroxide, and we leave it for 1 hour and dilute it, and added the 0.2 milliliter of chloride acid, and then we analyze it with the double beam spectrophotometers in wavelength 220 nanometers and 275 nanometers. And for the TP, the total phosphorus, we take the sample and add DI water and add some ammonium persulfate, and then we autoclave it for 30 minutes, and we dilute it and added the mixed reagent, and then the analysis of it using double beam spectrophotometer in 180 nanometers wavelength. Next. For analytical methods of algal species, the first one we do is the centrifuge the sample, and then we add some sodium hydroxide, and then we water bath it for 20 minutes and add the chloride acid for 1 milliliters, and then we water bath it, and then we extract it with N-hexane solvent, and then we trans-esterification using the sonic cutter, and we centrifuge the tube, and we move it to a sample bottle, and then we analyze it with GC mass. Next.
So the next one is algal breeding reactor design and operation. As is shown in a slide, there are operation and measurement. So for measurement is for TN, TP, TOC, pH, and algal mass. Next. And this is about TOC, or total organic carbon analysis. So algae samples were filtered using filtration paper, and then 1 milliliter of sample and 19 milliliter of deionized water were taken. The samples were then characterized using a TOC analyzer. Next.
And then pH, we use a pH meter, and then for blood flow solution for calibration, and immerse the electrode to the sample. Okay, next. This is our research design, and we use ESAN sample for A, B, and D sample, and we also use the first and the second sample from Blackwing Steel Lake at NKUST. Okay, next. This is the pictures of ESAN's algae samples. Next. then there are nitrogen concentration from Isans A, B, and D LG samples. Then make a repetition for A LG samples on different days, and each sample has different nitrogen levels. The first experiment for the average nitrogen from Isans A sample is 81.67 milligram per liter, and the second one is 42.3 milligram per liter. Sample B has a higher average when compared to sample D, namely 41.67 milligram per liter in sample B and 28.8 milligram per liter for sample D. And then the highest total nitrogen is the A sample, which is indicated the algae growth in sample A lower than the others. Okay, next. There are phosphorus concentration from Isans A, B, D, LG sample, and they also make a repetition for a LG sample, and each sample has different phosphorus levels, and then algae growth can also be influenced by other factors such as light intensity, temperature, pH, and the others, which support faster algae growth and reduce nutrient levels in the media. Okay, next.
This is the total carbon, total organic carbon, and inorganic carbon from Isan sample. The average of total carbon are 230 milligram per liter, and for total organic carbon are 121 milligram per liter, and for inorganic carbon are 109 milligram per liter. And for total carbon of Isan A sample are 217 milligram per liter, and for total organic carbon concentration are 137 milligram per liter, and for inorganic carbon are 77 milligram per liter. The organic carbon consumption by microalgae growth depended how much on nitrogen and phosphor concentration and nitrogen per phosphor ratio, especially when the phosphorus is sufficient. Next. So this is the black-winged steel-like sample we first take. Next. This is the second sample we take from the lake. We added some nutrients with 250 nitrogen ppm to the sample for growing the algae. We can see the change color to greener color. It's indicating the addition of nutrients accelerate the algae growth. Next. So this is the isolation of algae from the Isan sample and black-winged steel-like sample. The first one is isolation from the first sample of black-winged steel-like sample. The second is from Isan sample, and the last is from black-winged steel-like sample too. From this isolation, there are fungal contamination, and the algal colony is low. Next. So in another experiment, Yvonne take the sample and pour it to agar with a different volume, and we can see that there are fungal contamination too, and the algal colony may increase. But to eliminate the fungal contamination in the agar medium, we can use antibiotic to eliminate it. Next. So this is the nitrogen concentration and phosphor concentration of black-winged steel like before added the nutrients. We can see it's very low for growing the algae. And after 6 days added the nutrient, we can see the color change to greener color. It can be known that the nutrients can accelerate the algal growth. Next. And this is the algal from Isan sample, black winged stilt leg sample, and black winged stilt leg after the isolation. We can see the algae are spherical and green color, and this indicating the algae are Flora vulgaris. Next. So after the isolation, we do the cultivation. We do the Isan sample cultivation. We can see from the first one, there are no green color and then in day 80, the color of the Erlenmeyer is green, and this indicating the accelerate of the algal growth. From this sample too, this is from the blue color, there are no really small amount of the algae, and then after the added of nutrients, we can see that the algae really rapid on the growth. Next.
Okay, so the next one is chromatogram retention time. Data with multiple compound peaks was obtained from the GC-MES result to identify ingredient contents in algae, and each compound's fragmentation pattern was determined using the spectrogram data, and each compound structure can be determined by examining its basic peaks and fragmentation pattern, whereby x-axis is a retention time in minutes, and y-axis is total ion content or TIC. And apart from that, in the study, using 246 TBP for internal standard, which can help minimize the impact of both systematic and random errors during analysis. And it is almost all samples which show the highest peak with a retention time of around 20 minutes. And for Isan's B sample, is totally represented two main organic compounds with their respective retention time as shown in this table. Next. And for Isan's A sample, actually on the Isan A sample, there were 3 GCMS results with different times and methods. On September 15, GCMS analysis of 2 samples was carried out to obtain precise data. GCMS analysis in first Isan's A sample, represented totally 4 main organic compounds with their respective retention time as shown in this slide. Next. And GC-mass analysis in the second Isan's A sample represented totally 3 main organic compounds with their respective retention time as shown in this slide. Next. And for the third sample on October 9th, it is the result of the ingredient analytical procedure, which was explained previously. And the GC-mass analysis on this sample revealed presence of some bioactive compounds such as ketones and alkenes as shown in this slide. Next. So the last one is GC-mass analysis in the black quick stilt leg sample represented totally 3 main organic compounds with their respective retention time as shown in this slide. Next. So generally, all sample was performed the presence of fatty acids or their methyl esters as 3 main ingredients. The first main ingredient is hexadecanoic acid, also known as palmitic acid, is a saturated fatty acid with a 16 carbon chain. and it is the most common saturated acid found in animals, plants, and microorganisms. This compound has antioxidant, antibacterial, anti-inflammatory, and anti-androgenic properties. Also potential to biodiesel and can use in the production of soaps, detergent, and cosmetic. And the second main ingredient is 9,12-octadecadionic acid or linoleic acid, and is a polyunsaturated fatty acid which has possessed antimicrobial activity. It is also used in the production of soap, emulsifiers, and quick drying oils. As long as I know, there has been no further research regarding the application of linoleic acid as biofuel. And the third main ingredient is 9,12,15-octadecatrionic acid methyl ester or alpha-linoleic acid, which has processes antioxidant, anti-diabetic, and anti-inflammatory. Also can be used as a biofuel. And both palmitic acid and alpha-linoleic acid can be converted to biodiesel through a process called transesterification, which involves reacting the fatty acid with an alcohol, such as methanol, in the presence of catalyzed. Next. So here are the conclusion, and the first one is in the research, Chlorella vulgaris algae was successfully isolated and cultivated from both Isan's and black-winged lake sample, and this occurred because the algae medium gets greener with time, a sign of growing algae. And the second one, the algae can grow well after added the nutrients such as nitrogen and phosphorus and sunlight. And the third one, the result of GC-MS analysis show that algae extract from sample has three main chemical compounds, which are methyl ester of hexadecanoic acid, 9,12-octadecadionic acid, and 9,12-15-octadecatrionic acid that can be used as valuable materials and energy production. Next. So we do have a recommendation for further research. The first one is use the antibiotic ampicillin 3 hydrate for the isolation of algae in agar medium. And the last one is carrying out further research to employ an algae bacterium co-culture system for obtaining target substances and processing the extracted algae residues, becoming zero waste. Next. Okay, here are all the references that we used. Next. Okay, that's all from us. Thank you very much, and I will return to the moderator.
Okay, thank you very much, Anisa, Selvia, and Wulan for the presentation. That's a very interesting topic, and so there will be a question and answer session, and the questionnaires are invited to use the feature to raise hand. Is there any questioner? Okay, to the first questioner, Valia, please open your mic and.
Okay, thank you for moderator Kananda for the time. I wanna asking to Kanisa, Wulan, and Selvia. Yeah, before that, I want to introduce myself. I am Valia Rahma, chemistry student 2021. Okay, as a chemistry student who is about to enter the 6th semester or 3 year in college, I want to asking you all about that program exchange. So how the process requirement of the program you are following, and you can get an internship like magang opportunity in Taiwan. Do all of you enter from lecturer recommendations or apply personally? Yeah, maybe you can explanation further for me, please, because I am very interested about that, and I hope can be you are Kanisa, Wulan, and Selvia. Thank you, later.
Okay, thank you, Valia, for the question. Maybe Wulan, Selvia, and Anisa can answer Valia's question first.
Okay, I won't answer the Valia question. So for the process requirement for this internship, we received the information from Mrs. Fera about this internship program, and the file submitted should have been uploaded to on one of the website, but we were assisted by professor to apply this internship program.
Okay, okay, Valia, is there enough answer for you?
Oke, so berarti apa namanya lewat itu ya recommendation lecturer ya, not apply personal.
Yes.
Oke, thank you for that information, Kak Wulan ya, Kak Selvia ya. It's very helpful for me. Maybe I want to ask you a lot at personal chat. Can I do that?
Yeah, sure, it's okay.
Oke, thank you, Kak.
Okay, thank you, Valia, for the question. Next, Fazal Alifia, maybe you can open your mic and ask the question. Fauzan, or maybe we can next to the another questioner. Kinanti Istanti or Puji. Oh. Okay, maybe Puji. Can you open your mic? Okay. Okay, the question from Puji is Assalamualaikum. I'm so sorry for the asking through chat due to my unstable internet connection. May I ask for permission to give a question about Kaulan internship topic? My name is Puji Rahmawati from class of 2021. My question is, in the extraction of algae lipid, an ultrasonicator was used. So how does the sonicator work so that it can mix the ingredients in the solution? Maybe Wulan, can you answer the question?
Yes, okay. Thank you, moderator, and thank you for Fuzi. That's a good question, by the way. And for answer this question, so actually a sonicator is a tool used to produce ultrasonic waves, namely sound waves with a high frequency is above 20 kilohertz, which is humans cannot hear. And these ultrasonic waves are produced by a transducer inside the sonicator. And this transducer converts electrical energy into ultrasonic waves, which are then emitted into the solution containing the materials to be dissolved or mixed. And ultrasonic waves cause small bubbles in the solution, which then burst and produce high pressure so that it can break or mix the ingredient in the solution.
Okay, thank you, Wulan, for the answer. Next, there's a question from Kinanti. On the PPT, it mentioned that the disadvantage of using ultrasonicator extraction is the intensity needed to be controlled to prevent the side effect, if I'm not mistaken. So what are those side effects if we fail to control the intensity? Thank you. Okay, maybe Wulan, Anisa, or Selvia can answer Kinanti's question.
Okay, so maybe I will try to answer from Kinanti's question. So in my opinion, if what's the side effect, right? So I think the side effect is the the mixture is not really can pure, so it's still have many contaminants there, and it's not have a high purity.
Okay, Wulan, thank you for the answers. There's another questioner from Fauzan Arja.
Tes, suara saya sudah masuk belum, Kak?
Masuk.
Okay, thank you, moderator Kak Nanda, for time to asking. But before that, I want to introduce myself first. My name is Fauzan Alipsha, and I am in the 5th semester in chemistry department. Thank you for explanation, Kak Anisa, Kak Selvia, and Kak Wulan. But I want to ask about the material that you use in the research, that is chlorella vulgaris. Why did this study use the chlorella vulgaris? And then if we want to change the materials, which is the chlorella vulgaris, to another materials, is it possible to change it or not? Maybe it's all for the question. Thank you.
Okay, thank you, Fauzan, for the question. Maybe Selvia, Anisa, or Wulan can answer Fauzan's question.
Okay, maybe I want to try to answer the question. Thank you, Fauzan, for the question. Maybe I will explain a little bit about microalgae and then chlorella vulgaris. For the first, microalgae are rich in lipid, as we know for our presentation before, and however, the lipids of algae can be altered by varying the parameters such as light intensity and then carbon dioxide concentration, temperature, and nutrient levels. And then green microalgae, especially chlorella vulgaris, was isolated from industrial effluents, and the algae's lipid were then removed and turned into biodiesel. And this process produces the highest output of chlorella vulgaris biodiesel ever measured at 87.6%. That's a high presentation for chlorella vulgaris. And your question before, how if we change the microalgae, it's can possible or no? That's possible to change the microalgae for different microalgae such as Ancritos species, and then another microalgae species. Okay, is that clear for Fauzan?
Anisa, for the answer.
Okay, thank you.
Okay, thank you, Fauzan, and all the questioners. Maybe we can move to Prof. Chen or Dr. Fera. Perhaps is there any question or response, please?
Okay, maybe I have one questions. How much the lipid yield obtained during the isolation process? Presentase lipid yang diperolehnya berapa?
Maybe we didn't have information about concentration lipid.
Uh, yeah, compared to the alga extract, the yield of lipid obtained compared to alga extract, the percentage. Is there any information?
I think there's no information because we extracted from the algae and then we analysis with instrument for analysis the chemical compound in the algae.
So the quantity of the lipid hasn't measured yet.
Yeah, that's right.
Okay, so we can see the whether we can scale up the process to obtain the lipid yield in high amount. Okay, maybe from Prof. Chen, is there any recommendation?
Well, actually, I do learn from some questions. If that's okay, I might try to answer some of the audience question as well. I think for the last first, I think Professor Fera is a very nice question, but I think that's the design of the study for now. Okay, the original study we want to search for the native algae which can produce lipid or some other valuable ingredients. Okay, so now we are in the search process. And we do know that once we find some kind of algae, we need to have some kind of operation or the reactor operation, and we need to modify some kind of operation factor before we can get some algae that can actually produce some meaningful amount of lipid or some other substance. So this is kind of different stage of the experiment yet. So we didn't really try to focus to measure the percentage of the lipid at this moment. Okay, because we know it's low, but based on the gas chromatogram and the mass spectrum photometer, actually we can know the pretty much the ingredient in that particular algae and see if they have certain kinds of lipid in there. So we might have a chance to incubate those algae in the future. Okay, so the answer is no, we don't know the percentage yet for Professor Fera's information. And the other thing I think is very important is that once we know they have certain ingredient in the algae, then we have to kind of extract them very carefully. And then in terms of meaning, the meaning of carefully is that for the sonication, you know, it's very, we can adjust the intensity of the power or the intensity of the sonication. Okay, so two things we are worried about. One is that if the intensity is too low, then we got nothing. That everything will contain still in the cell because that algae has a cell wall. It's kind of a strong boundary for a cell. And then if we got too strong, that means we will mix everything together, including the lipid and their organelles. And then we will mesh up everything. Maybe we can put it into a certain kind of like we making the salad dressing. Okay, we mix the oil too fast and everything become soluble again. So that's because we say that if we do too little or too strong of the sonication, it won't be good to extract the maximum amount of lipid. Okay, that's why we try to test the intensity of the sonication. So that's so far I remember the question from the audience, two questions. I think I can give you some explanation for the study, but Since I hope I have a couple minutes, do I? The mediator, can I have a couple minutes? I would really like to talk about this internship program from Anisa, Wulan, and Selvia. And I personally kind of to say something that I really these three actually are dream students. Okay, I think they have very a nice understanding in terms of the whole project are going to do, and actually they make a very nice explanation and data collection. So I think they fully understand what we are going to do in the next couple of years. And actually this LG study is a project from the Ministry of Agriculture of Taiwan government. Okay, they've been support our laboratory for almost 6 years. And we really want to do something on this particular project. It's kind of research type of project. So we still have maybe we have another student working on it. So maybe we are working on it for another 2 years on this study. Okay, and then also I think they are very nice observer, and also they make a very good data recording job. Okay, and then I think I listened to their PowerPoint many times. Okay, even they were here, and then I still kind of impressed every time they present the material. Okay, they kind of are doing a very nice job in there, and then and we do have other moments except that the study work. Okay, I think I do remember I went to the airport and picked them up and at the middle of the July, and then after that we have the student meeting and we explain the whole thing to them and then ask them to collecting the information. So everything for them, the three, is from the zero. Okay, they don't really working on LG before, and then I'm glad that they kind of help us to get started on the lake LG. Actually, we have another student working on water reservoir LG. So this is kind of we take sample from many places, and we have students to schedule to take many sample from the water reservoir around Taiwan, maybe in other north and center and the south. And the reason we do that is we really like to find some local or native species of algae. So if we need to cultivate them, then that means that the Taiwan environment will be suitable for them to grow in a massive amount, something like that. Okay, so kind of enjoy to have them here. So that's only one thing I feel regret. That's they just stay too short. I wish they can stay a little bit longer so they can see how the full project will going, and then maybe they can get some more detailed information. And actually, they make really nice recommendation as well. We find out we do have some kind of material problem. So actually, we already have an antibiotic, and then we test the antibiotic just recently, and it works just fine. So I think we can use antibiotic to control the bacterial growth while we incubate the algae. So I think that's pretty nice observation, and it's in Anisa's report in the suggestion section. Okay, and then I also have one expectation that the student is very nice. So maybe someday we still have opportunity to cooperate for something, okay, in the future. Okay, and then also one information I would like to review to the students, because I think some students is asking about TEP program, and this program actually is it's actually a huge program. Actually, it's supported by the Ministry of Education by Taiwan government, and we have a website called TEP website. Okay, and then if you go onto the website, you can see all kinds of experiment, laboratory, and then they do all sorts of research. And the New Year's program, the TEP program, and then my center also get the quota for that. So that means we can accept the students again from the university out of Taiwan. So that's what we just got the information that we do get a quota for the 2024. So I'm preparing material. Maybe I will share my program to Professor Fera, and if your student too interested in the program, we can kind of try to find someone to continue the TEP program with your university, okay, or your college. Okay, I think that's short, but I think the time's kind of late. You have 1 hour limit for this meeting. I think it's kind of a long out of time, so I will end here. Okay, so once I pass on the program, I pass on the TEP program. I would like your help, Professor Fera, and maybe you can deliver the information to the students. Thank you very much.
Thank you very much for the opportunity given to our students, Prof. Chen, and also providing the funding also. So we hope in 2024 there will be our student will join this program. Okay. Okay. Is there any questions from the students? So it is available for our student to apply the program. Where is the due date? When is the due date?
Is that the schedule for TEP program, Mrs. Fera?
Yes, the time for our students to apply or to join this program is from after July. Is it possible or not? Since you still mute, your microphone is off. Prof. Chen, your microphone is off.
Sorry for the. I mean, the project time is the year of 2024. So the student just come in that the time frame, which is fine. That means come in this year from January to December. Okay, and then the stay, I think it's pretty much from 1 to 3 months. Okay, that depends on the student. Pretty much like the I think the Anisa run and Selvia's program. But I will give you the detail. I have flyers. So because the project I just notified that we just got the vacancy to recruit students. So I haven't have that finished yet. Okay, I will prepare that. So you are maybe the first one know that we do have the vacancy for students for the TEP program. Okay.
Thanks for the information. Maybe we can follow up. Yeah, your program then. Okay, Ananda.
Okay, thank you to Prof. Chen, Dr. Fera Kurniadewi, and all students. We are already at the end of the session. Thank you to everyone for attending this seminar today. I'm Ananda Indah as the moderator. This is all for me. Thank you so much. Saya kembali lagi ke bicara. Terima kasih.
Okay, thank you.
Okay, thank you very much, Prof. Chen.
Thank you. So, Professor Fera, do you say you want to save some time to talk about the other application?
Yes, maybe Fauzan would like to ask questions about the program. Fauzan?
Tes. Suara saya sudah masuk belum, Bu?
Ya.
Hi, nice to meet you, Frozen.
Nice to meet you too, Professor Chen. Okay, maybe I want to introduce myself first again, Professor. So my name is Fauzan Alipsyah, and I am in the 5th semester in chemistry department. I have read about the program of IIPP, and then I'm so exciting to join that programs. And then I have registered to the website, and maybe I have some question about the website. So the first one is about passport, Professor. In the website, I have to upload passport, and then what time should the passport be processed? Because in the website of IIPP programs is required to upload. And then about certificate enrollment, and also one letter of recommendation. Where do I can get those files? Whether in campus or in Taiwan? Maybe it's all my questions. Thank you.
Okay. And the second thing you talk about, I didn't get it. You mean in a certificate of enrollment? Is that what you
Yes,
said?
yes, certificate enrollment.
Okay. No, let me explain a little bit the program to you, because this program is really a very specific aimed for certain researcher in this program. It's supported by the National Science and Technology Council. Okay, and then it's a different program from TEP. And the program actually, they have a fixed quota. So the total, I think the program, the IIPP, they only will support 600 persons all over the world. Okay, so they if, for example, the Frozen, if you apply, then the quota will be 599 remain. Okay, so now everybody is kind of try to apply for the program on the website. So that means if the quota use up, then everybody just have to wait for next year. Okay, so that's the game to play. So if for me, just fill out the form on the website, and then go ahead find my information, because every professor we already registered as a mentor on December last year, maybe even earlier, November last year. So the program already set. So the competition is free. It's based on when you got matched, and then when the system process you to agree, and then they will count you as a vacancy field, something like that. Okay, so now I received more than 50% apply for the project already. So the thing is, you have to fill in the information on the website. I cannot really help you on that. Okay, you have to fill in the information on the website. And then after you've done all the information on the website, you go ahead and find my name, and then you select me as a mentor, and then the system will notify me to pick you up. Okay, so that's the way to, and then we will get a match. And then once we have a match, then if you say, okay, then we will send you a term that have some kind of agreement, and you say, okay, then the system will process your application all by themselves. Okay, and the only question you mentioned is that actually, I don't think you need any certificate of enrollment. You don't. Once the program accept you, that's an enrollment certificate. And then for the passport, I think I received some student, they fill in on that particular request. just by saying that it's applying. I am applying the passport. Okay, and once I got the number, I will upload my number. Okay, and then if they need certain kind of picture file, you just send them a picture file, maybe just write something and then say I'm applying the passport. Okay.
But the requirement is the student have to upload the certificate enrollment, and then yeah.
So why is the certificate of enrollment? I don't know, actually.
Yeah. Is
Do you have
it
any-
kind of a letter of recommendation? But I think there is also a letter of recommendation in the next uploaded document.
Mhm.
So maybe we already we don't know what kind of certificate enrollment here, and then
Mhm.
yeah.
certificate of enrollment?
Fauzan, can you share the screen?
Yeah, if you can share me. Sorry about that, because I am in the mentor side.
Yeah, yeah. Maybe
System.
it's different look.
So I don't really know how the applicant sends
Yeah.
the information. Okay.
Fauzan, can you share the?
Okay. Maybe for the website.
I know. Maybe that's the certificate to show that you are a student in the
certify that he's a student of our study program, right?
Yeah, that's right. The student of your university. Yeah.
Okay.
Not from me. I think that's from the student side. Yeah.
Okay. There's also uploaded attachment including a catalog of literacy work. Patents. The last one?
The last one?
Yang ini nih. Yeah. Upload attachment.
Okay. See the status, the red star. Okay. If the item has not the red stars, they are not required material. You don't need that.
Okay.
Yeah.
Okay. So there's also a statement, the red one. Coba ke atas nih Fauzan. Please complete the matching with before 1 April.
Period time of each. I think it's fine as long as you can send that information out. We can change the time.
Okay.
Okay, when we try to match the mentor and the students. Okay, so just put on some date. They can
adjust the time.
That's right. We will adjust the time when we match together.
So, Fauzan right now processing the passport, right? How many days?
Yeah. And then also, I think he is talking about matching time, not really the time you stay in Taiwan. The matching time means now.
To apply.
Right, to apply. That's right.
Before the 1st of April.
That's right.
Okay.
Okay. So the father and I will kind of encourage you to just fasten up the process, because as I mentioned, the quota was taking away every day. Okay. And then if they use up all the 600 quota, the program will be terminated. Something like that.
Maybe Fauzan will finish all the requirement document in this week, yeah, Fauzan?
Okay.
Okay.
For the
Okay.
passport, just as I suggest, just say I am applying, and I will upload the information once I got a passport. And if they want a photo file, you just photo that sentence.
Oh, okay.
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 Mhm.
Okay. Okay. Thank you, Prof. Chen.
No problem. So that's about it.
recommendation.
Okay. And I will say thank you for Anisa, Wulan, and Selvia for your
Oh.
work in Taiwan. And I hope that we might able to kind of work together sometime. Okay, in the future.
Okay, Anisa. Anisa and friends want to say something to Prof. Chen?
Well,
Anisa?
not necessary right now. I think we have a 9 group, so we still can contact each other from
Oh,
time to
okay.
time. Yeah.
Okay. Thank you very much for your time and
Thank you.
your recommendation. Prof. Chen will contact you in email if there is a problem. Okay.
I do have another information. Maybe you guys, I don't know if anybody interested. That's for invitation for professors to come to Taiwan. Okay. So Professor Fera, if you know, just know that if some professor would like to come to Taiwan and maybe give a short presentation about your research.
Or maybe Prof. Mukti. I will ask
Maybe
Prof.
if
Mukti
you
to.
can
yeah.
kind of ask for me privately, see if any professor would like to do that. Okay, we do have a program that we like to invite professors to come over.
Okay.
And then I think they will pay at least the plane ticket.
Okay. Okay. Thank you.
Okay.
Thank you.
Okay, thank you, then. Okay, see you then. See you guys. Bye bye.
Okay. Have a good day.
You too. Bye bye.
Thank you, Professor. Thank you.
No
Thank you
problem.
very much, Professor
Thank you
and
very much
Bu
for
Fera.
Miss
Bye bye.
Fera.
Bye
Bye.
Bye.
bye.